So thank you very much. Thank you very much for joining us today for our microelectron diffraction webinar. We're excited to bring a number of people from the Australian and the regional electron microscopy community together and to present some of the recent capabilities in this space to you all. So microelectron diffraction is a technique that allows for fast, high-resolution structure determination of small molecules and proteins. Atomic details can be extracted from individual nanocrystals, smaller than about 200 nanometers in size, even when they're in a heterogeneous mixture. The data is acquired generally on cryo-transmission electron microscopes, using electrons as the incident beam. Since MicroED is a diffraction technique, samples need to be crystalline. The crystallization process is generally the same as for X-ray crystallography. However, much smaller crystals can be used in MicroED because the interactions of the crystal with the electrons is much stronger than it is with X-rays. Crystals that are only around 100 nanometers in size can readily be analyzed, and this may significantly shorten the sample preparation process and allow for the analysis of crystals that are too small to diffract with other methods. Data collection is completed in only a few minutes, and three-dimensional structures can be determined at atomic resolution. So today we're joined, firstly, my name's Ryan Shaw. I'm the manager of the electron microscopy business in Australia and New Zealand for Thermo Fisher Scientific. And I'm joined today by some colleagues, both from Thermo Fisher and from the Australian scientific community as well. I'll just introduce everyone quickly. First, we have Dr. Hongyi Xu. Hongyi is currently based at the Australian National University and works on the development of electron crystallography methods for crystal structure determination of inorganic materials and small organic molecules, as well as peptides and macromolecules. We also have Dr. Wim Zhong, who is one of my Thermo Fisher scientific colleagues. He's a structural biologist by training and received his PhD from the University of Edinburgh. He has over 10 years experience in structure-based drug design with diverse working backgrounds in university, research institutes, protein biotech companies, and drug discovery companies. His last position was Director of Protein and Structural Sciences in an MIT Harvard spin-off company, applying cryo-EM and crystallography for cancer drug discovery. Currently, he's the Sales Development Manager in Thermo Fisher Scientific, supporting the electron microscopy pharma business in Asia Pacific. Next, we have Dr. Boyin Liu. He completed his PhD in 2015 at Monash University. During his candidature, he studied and developed 3D imaging methods for cellular imaging using tools including TEM, FibSem, and AFM. He then moved to the University of Melbourne for postdoctoral research on malaria diseases. Since 2019, he's been responsible for cryo-EM training and application support throughout Asia. Next, we have Dr. Yufeng Zhao, who's the Senior Technical Officer in the Fluorescence Analysis Facility and the cryo-EM facility at the Molecular Horizons facility at the University of Wollongong. And lastly, we have Dr. James Bauer, who's the Director of the cryo-EM facility at Molecular Horizons at the University of Wollongong. Really happy to have you join with us today, and I'll just hand over to James for a little bit of an introduction about the facility at Wollongong.
So thank you, Ryan. It's really nice to see all of your lovely faces out there. So we're the University of Wollongong. We're the place with the funny name, but we are very serious about cryo-electron microscopy and developing new techniques. I'm really excited to be on this discussion here around microelectron diffraction, which is something I've been very interested in since about 2017 when I was at the University of California, San Diego, and Tamir Gonin and his group came out with this new technique, which was extremely exciting. where you could actually see hydrogen atoms in your structure, which is much more difficult using X-ray crystallography because the scattering cross sections are so much lower. So it's kind of started initially back then, and it was such a challenging technique. Everybody told me just not to do it. But after coming to University of Wollongong, I picked this back up again, and I had a couple honor students start working on this technique. We put it on just a basic machine, a T12, just to see what we could do, just using a standard camera. And really, a couple years later, Yufeng came along, who you'll hear from later, working with Dr. Aaron Oakley. And we started pursuing this technique about 3 years ago pretty heavily. Tamir Gonin came out to the University of Sydney. We had a meeting with him. I sat down with him at the microscope, and I really have to thank him. He spent a lot of time showing me how this technique works and how to acquire the data. Of course, that was passed on to Yufeng, and she has taken the ball and is running with it. And I'm expecting we're going to score a lot of goals. So the University of Wollongong cryo-EM facility is an open, outwardly facing organization. So we have customers from the pharmaceutical industry. We have customers from across Australia, New Zealand, customers in the US, and so forth. So looking to expand our customer base. We are now have a working workflow, and we have some techniques for getting this done, all kind of done really in a manual way. So we're really excited about working with Thermo Fisher on the new tools, the new software, EPUD, and getting some of the new camera systems, some of the new hardware to make this possible. We have a completed and working workflow base both for small crystals, for drugs, and also for larger proteins. So we're available and looking for customers who would like to work on this technique and help us develop this further and break some new boundaries in molecular biology.
Thanks very much, James. So without further ado, I think we have a pretty packed schedule for the afternoon. I'll pass over to Hongyi for his discussion of the potential impact of MicroED for the discovery process.
Okay, can you hear me now? Am I muted? Can you hear me?
Yes, we can hear you, Hongyi.
Okay, great. Yeah, thanks for the introduction, Ryan. It's my great pleasure actually to give this talk back in Australia again. So I joined ANU about 2 weeks ago. So I'm still learning all the bits and pieces and going through all the inductions and safety trainings. Right, so actually all the work I'm going to present today I actually done with people at Stockholm University. So hopefully soon we'll establish a MicroED workflow at ANU also. Right, so I was given the topic actually to talk about how to apply or use MicroED or 3D ED as a tool for the pharmaceutical industry. Right, so before I start, as I said again, all this work were done by people who are working at Stockholm University, so this Chardon's group. I was a group leader within the large group, looking after about 10 people, basically developing 3D ED, MicroED, also serial ED methods for studying small molecules, peptides, and also protein. So my main interest is working on protein because these are more challenging to work with. It's difficult to grow and difficult to prepare cryo-EM sample of these protein crystals, and they are much more fragile compared to small molecule. But then on the small molecule side, we're trying to make everything a little bit quicker, easier, and more accessible to people who are not so familiar with the method but would like to try this method. Yes, so there are a lot of close collaborators we have also at Stockholm University, the biochemistry department. So Martin's group has been very fundamental. Our key collaborator actually the first group I worked with on protein crystals, and Hugo was one of Martin's postdocs. And Max Klabers, he's now with Tamir Golden at UCLA, so he was my postdoc who did a lot of great work on the micromolecular side of things, so working with proteins. I believe he's moving back to Denmark very soon. And Jing now is at Rosalind Franklin, so she did a lot of sample preparation development for cryo-EM, MicroED crystal samples. And then also SciLife Lab, which is the Swedish national cryo-EM facility, which I'm really proud working with. All the method we develop actually go directly to the cryo-EM facility as a service. So this is a similar model we're trying to establish here in Australia. Hopefully, a lot of the microscope labs can already be providing MicroED service while we take the research side of things and then develop new methods. Once these new methods be developed, this will be available at these cryo-EM facilities or microscopy facilities. So why do we want to use 3D MicroED? The way I see it is a complementary method to all these existing techniques, including X-ray, neutron, NMR, SPA, or cryo-ET. Right, that's not going to solve all the problems you have, but it's going to be a new tool or new option that you can use to look at your structure-related questions. And as Ryan mentioned, and James, that why do we use electrons? Because they interact with matter much, much stronger. So much smaller crystals can be studied. If this is the crystal we use for single crystal X-ray diffraction, this will be the crystal size we need for MicroED. So anything considered as powder in pharmaceutical research or in materials research can be treated as single crystal for MicroED. In the protein world, you usually grow these proteins in the drop, and then these large, nice, pretty crystals are the ones you're looking for single crystal diffraction measurements at synchrotron, usually. And then these needle-shaped small crystals are actually perfect for MicroED. So about 30 to 40% of the plates you used to throw away contains crystals that is might just be suitable for doing MicroED. Right, and also electron interacts not just with the electron cloud, but also the nuclei, so it's possible to probe charge. And we actually spent the last 4 or 5 years on this topic, and it actually turned out to be much more complicated than we thought. But then electron should still be able to probe charge. It's just a little bit more difficult than what we used to understand. And it's also dynamical because the strong interaction, right? So when the electron goes through the sample, it gets scattered multiple times. Then the intensity we measure at the end is not so accurate comparing to X-ray. So this was a hurdle before that people, especially the physicists, say that you cannot use electron intensity to solve structures because of the dynamical nature of the scattering. Right, but nowadays, especially Lucas Palatina's group in Czech Republic has developed this dynamical refinement procedure, so that we can treat the intensities we measure and solve the structure and calculate the dynamical intensities from the structure models and compare with our experimental data. So this makes everything much more accurate. And then by using the dynamic refinement, we can actually tell the absolute structure of the molecule. So this will be very useful for the pharma industry also. And last but not least, electron can be focused, so we can take high resolution images, right? So with diffraction data, we get the average structure of the crystal, but then we can use high resolution image to look at local disorders and local information of the crystal. So this is a very great combination between imaging and electron diffraction. So in all, this is called electron crystallography. Right, so I wouldn't go through too much about how to do MicroED. You're probably more familiar with this. So it's continuous rotation of the crystal and continuous data collection at the end, but we're collecting data in reciprocal space, so we're measuring the diffraction pattern. And then we reconstruct the patterns into the 3D reciprocal lattice, and then from this lattice, we extract or integrate the intensities from each indexed reflection spot, and then generate HKL file. Then we can run the X-ray softwares to do structure determination. And nowadays also running YANA, which can do the dynamic refinement. Right, so I was asked to talk about how MicroED can be used in pharmaceutical industry. So I see it as two ways, and I've been collaborating with companies like AstraZeneca in both of these aspects. So at the very early stage of the drug discovery, what people can do is to do this so-called fragment screening, right? You have your target protein, and then you crystallize it, have the apo crystal that diffracts better than 2.5 angstrom resolution, and you start to soak small molecules into this protein crystal to see which molecule you soak into the crystal would actually make a complex with the protein. And then if the complex is inhibiting a certain functionality of the protein, then this can be used as a potential drug molecule. And then you can grow the molecule even larger to have a better affinity um, to form the complex. So this I will not talk about so much in this talk because this was more interested, I think, from the audience. That's what I was asked. So we look at how to use MicroED to look at very end stage of the pharmaceutical development process, is to look at the polymorphs, which is the different packings of the same small molecule, and also look at how we can look at co-crystal, for example, and look at all these different crystal structures and the properties of the crystal using MicroED. And basically, we can do phase analysis. We can look at we can do phase identifications. So if we have a powder of mixed polymorphs, we can take this powder and then treat each small crystal as individual single crystal, and then do phase identification. And if it's a new phase, we can even do structure determination. Right. We also want to do some quantitative analysis. I put the star here because this requires very rigorous or robust sample preparation and data collection protocols. So really, before we can go to high throughput, quantitative analysis will be quite difficult for MicroED because we only look at very small amount of the sample. So powder diffraction is probably still better to do this task for now. But later, with the high throughput and automation, I think we can achieve this also with electron diffraction. And then we can also detect minor phases in the powder. So this is very useful for IP protection. Yes. And then we can look at drug expiration date because we can look at the same powder over time and then see how the polymorphs are changing and then transforming between each other. So one phase transform into a different phase over time. And we also can look at the I'm also going to talk about the stability and robustness of the workflow we have at the moment. Right, and also I want to sneak in just a little bit some of the new methods that has been developed in the lab in Stockholm, hopefully soon at ANU also. Right, so for powder sample, it's actually very simple. We take a powder, a pharmaceutical compound, and then we just crush it and then deposit onto the TEM grid. Right. So usually what we do nowadays is to actually do a PXRD screening first to see if the powder is crystalline. And this also gives us the expectation of how much resolution we can get from the powder. And it's a very good quality control also. So if something is sensitive to the vacuum in the TEM, right? So what we measure at the end should match what was in ambient air. For example, so we can use it to compare if a sample has been modified by a procedure we do during the MicroED process, right? And as I said also before, crushing and depositing the sample can bias the small crystallites that ends up on the grid. So this I wouldn't go into too much detail. So for any quantitative analysis for the sample preparation part, we also have to be more bias-free in a way. Right, so one of the examples, first examples I would like to give is this drug that treats skin cancer. So I think for any one of you who's been working with pharma compounds, when you want to grow new polymorphs, it's very likely or very often you see this kind of spherulites, right? So you can get powder patterns from them. They're beautiful, but they're very difficult to solve structures from. So what we do with MicroED is we can crush this spherulites and then end up with these small fragments the spherulites. And often these small fragments are single crystal. And by using MicroED, very straightforward, we can collect very nice data from these small fragments from these beautiful spherulites and then solve structures and treat them as if they are single crystal. So it's very straightforward for this polymorph discovery. It's actually very useful. It will speed up the process that a polymorph can be discovered. Right, and the next example I want to show is discovering a new phase. So this is indomethacin. It's a non-steroid or anti-inflammatory drug. So for 47 years, this drug, the structure of this polymorph was unknown because single crystal growth was almost impossible. It's very difficult to grow a large crystal out of this compound. But there are actually been reported there are two ways to grow this crystal. And one is using solution-based method, and one was using the melt crystallization method. And then when people look at all these characterizations of these two different, they believe they are the same polymorph, but then two different growths. But if you look at all these characterizations, they are very, very similar. But are they really the same phase? Now we know they are different phase. If you look at the powder, there are subtle differences between the powder pattern, right? So what happened is we took the solution-based crystal first and solved the structure, and around the same time, actually, Mauro Gemmi's group in Italy solved the structure of the same polymorph. And at the same time, Molly was also working with the crystal polymorph that was grown with the melt crystallization, believing to be the same form. And then she actually got a very, very different unicell from this crystal. And then we realized that there are probably two different polymorphs. Right, so actually it took a very long time to solve the structure of this polymorph because the large unicell and the disorders in the structure. So only with MicroED we could look at the small crystals and actually distinguish these two are different polymorphs. And this was a question that was unknown for 47 years. So that was quite nice showcase to see that detecting for polymorph discovery and also detecting new polymorphs in pharmaceutical compounds. Right, and the next experiment is a little bit more creative. So we were trying to see if we can crystallize a certain compound of a small organic molecule and then crystallize it on the grid and then follow the crystallization process with MicroED. So we apply the solution of the compound and then incubate it for different amount of time, blot away most of the liquid, and then freeze the grid to look at what was trapped in this time point during the crystallization process. And this was after 20-second plunge freezing. This is carbamazepine. It's a very simple sample to work with. I think a lot of people use it as a test sample, but the most stable phase is the carbamazepine dihydrate, right? So after 20 seconds, we don't see any crystallization actually started, but after 30 seconds, when we plunge freeze, we actually see 3 intermediate phase that was not known that will happen or form during the crystallization process. And then at the end, if we wait long enough, finally the crystal becomes the dihydrate. And this is also done with cryo, so you can see the water molecules were also refined in this structure. So MicroED could be also very useful for doing this to understand how crystals form and then to look at all these intermediate states during the crystallization process. The last example of small molecules I would like to give is a very recent work. It's kind of related to the phase transformation of a drug over time, right? So this is an inclusion complex of antifungal drug with PEG. And the reason why our collaborators do this is that it can modify the properties of the pharmaceutical compound, of the crystals, so the solubility can be improved by introducing PEG. So what they did was they grew this crystal with melt crystallization method, and they thought this was a single phase. And the powder X-ray diffraction suggested it's a single phase. And then after 10 days of transportation with FedEx, so they're actually doing the synthesis for us. And actually what we got was actually a phase mixture. So when we did the powder on this spherulite, the powder pattern actually changed from a single phase to multiple phase. And then we thought it's a perfect sample for doing for testing a high throughput protocol we've been working on. So our group is developing softwares to do high throughput MicroED, fully automated, and at the same time, Thermo Fisher has this EPUD. So this work was actually done with EPUD, but with manual crystal picking, and each crystal was diffracted over 80 degrees of tilt. So each data set is pretty much complete for structure solution if we want to. So over 2 days, we collected 230 sets of data. And then out of this 230 data sets, we actually identified 6 different phases. So 3 of them we could see from powder, 2 of them were hidden, and then 1 was actually a very, very minor phase. No, sorry, form 2 was a very minor phase. And then within the powder, we also see the original form that our collaborator has grown. So this is the new polymer of a drug polymer inclusion complex. Right, so I'm putting manual crystal picking here in red because, as I said before, if we really want to do phase analysis, we have to get rid of the ambiguities in picking, right? This is time consuming, and also it introduces bias because as human, we usually pick on things that looks pretty and that we think is going to diffract really well, right? So we really want to get rid of this and then look at everything that is on the grid. First, make sure that what we put on the grid represents our sample, and then we look at everything across the grid to make sure we don't have any biases. So until then, we can start to do the quantification. Right, so if we look at the data processing, it's automated through a script-based software that was developed by Steph Schmitz, one of the postdocs in the group before. And then all the data were processed automatically and clustered automatically. So from this cluster analysis, you see, because the unit cell that were quite accurate, it was very easy to cluster out these 6 different forms just by looking at the unit cell parameters. And then if we look at the powder XRD, the comparison, if we just put in form 1, which is the dominant phase, we see that there are several peaks that were not indexed. So this is indication of additional phases. Then we put the inclusion complex 1 phase in, and then there's still a few peaks not identified. But then we put in form 3 again into the refinement, and you see that most of the peaks were actually matching with the experimental data. So this suggests that if we do it with powder diffraction before, only 3 phases would be discovered. But now with MicroED, we can look at small quantities of and also minor phases in the mixture. We found 6 different phases. And as I said, this is a script-based software. So now there's a new software written by my PhD student, Lei. It's called Automatic Low Effort Integration, so AutoLei. This is going to come out very soon. So this will be compatible with a lot of different machines, and then you just have to do calibration once. And this also does data processing on the fly. Right, so what we want with the workflow is we want to have a robust sample preparation, as I said, rigorous data collection protocol, fully automated, non-biased. We can do serial ED combined with MicroED, right? And then we can do this automated data processing, hopefully on the fly, and then do phase analysis automatically as well. So this is what we want, but this is still very expensive. So when I talk with pharma companies, when I visit them, they actually say they rather outsource their MicroED work to experts because it's still very expensive to them. So I think it's a very good opportunity because Thermo Fisher is here. So I think the best machine for the pharma industry will be just taking the powder, dump into a electron diffractometer, which was generated by ChatGPT. I don't think it's as complicated as this one. But then the data collection and sample preparation would be taken care of fully automatically. And after a couple of hours, you should be able to get a cluster. And if there's a new phase, you should be able to get the structure of the new phase as well. So a machine like this and be in line with the current process of pharmaceutical producing pipeline will be very nice, I think. Okay, so I'll squeeze in a little bit more method development, which are all unpublished results. So first, I will talk about environment sensitive sample. So this is a sample which we received that is very sensitive to the humidity in the air, and it's also vacuum sensitive and beam sensitive. So what do we do then? We have to make sure that the sample does not see air during data processing, during the sample preparation, right? So we actually borrowed a glove box from Martin's group, and then we crushed the powder in the glove box in nitrogen environment. And then for this glove box, there's a special attachment, which is a cold trap. So we can actually have solid ethane transferred into the glove box and being kept cold before the plunge freezing. And then we can take it out and then defrost the solid ethane to make liquid ethane, and then plunge freeze our grid into the liquid ethane, and then cryo transfer the grid out of the glove box. So this was done for small molecule, but the purpose of this machine, actually Laura is working on, is to trap the intermediates of a redox reaction in an enzyme. So we're actually studying enzymes in different states, oxidation states, and then look at the charge of the metal ions in the enzyme. Right, so this was within the glove box. We can see beautiful diffraction. And then if we do it in air, you see that even the crystal morphology changes very rapidly, and we don't see any diffraction, obviously. So this is one way to actually work with very environment sensitive samples. So for lithium-ion battery, I think this will be a very nice workflow. And we can also now work with samples in liquid. So this is a collaboration with a group at DTU, Christian's group at DTU, and also they commercialize the chips nowadays. So these are nanofluidic channels. What you're looking at here is these are the silicon nitride film glued together. And then in this channel, there's about 200 nanometer depth, and the liquid can run through this liquid channel. So this crystal you're seeing here is actually submerged in liquid. And in this case, the crystal is in air, so there's no liquid in the channel. But in this case, the channel is filled with liquid. So with this, we can study crystals in different chemical environment. And because of diffraction, different to imaging, right? The background you get from having the liquid is not so severe compared to imaging. So we could solve the structure very nicely using the crystal submerged in the liquid using these chips. Right, last but not least, we also are interested in high resolution protein structures, right? In 2022, Tamir's group published a very nice workflow using very advanced instruments. So they were using FIB milling to mill crystal lamellars, and then use a direct electron detector for diffraction data collection, which is a bit overkill in my mind, because these are very sensitive and reads much slower compared to these hybrid pixel detectors, for example. So we're trying to make it easy and simple for everybody. So basically everybody now having the cryo-EM lab can do this. So we do the crystallization method by using beads and seeding procedure. So we seed make a lot of seeds of a small lysozyme crystals, and then we're adding a little bit more lysozyme at the end to recover the fragments. And then because there are a lot of seeds, so the crystal don't grow very large, and the protein are used up very quickly. And then we do hand blotting, which is a new procedure, and then we use scintillator-based CMOS detector. So this was what Lei could get. So this is a Titan Krios with CTDs. I think most of the cryo-EM labs should have this already, and then we can get to 1.0 angstrom resolution as well. And the map is actually slightly better than the published one. So if we look at the map, even disulfide bridge, because we can merge a lot of data sets using auto-lay, and we don't see much damage even around disulfide bond. And if we look at this alpha helix, we can see all the hydrogen signals coming out in the hydrogen bond. Right. And the last but not least, cryo-ED, which is something we're very much interested in at the moment. So I think it's also very capable to be implemented on the cryo-EM or also TEM out there. So this was a work Max and I did in 2020. We can show that it's very clear to have the poles of the drug molecule binding with the protein, right? So with cryo-ED, what we can do nowadays is we collect one diffraction from one crystal, so we don't have to worry about beam damage, and we sample tens of thousands of crystal within 10 to 20 minutes. And then we can combine this data set to create a 3D data set that is similar to what MicroED can get. So the difference nowadays is we cannot just tell that the ligand is there. We cannot just tell the pose of the ligand. We can look at the difference of two very similar ligands in the same binding pocket. And then we can even look into some of the chemistry now because of the resolution and the damage-free nature of these structures. So we are in the process of publishing the cryo-ED work. We are very excited about this. I think this will be adopted in many labs in the future. Okay, so finally, MicroED did a lot of capabilities in Australia about MicroED already. So the first dedicated machine was actually built and installed at UQ. So this is Glaciers 2. And the Kasunis Nama is responsible for this. And I think they are already collecting very nice protein crystal data sets using this machine. And also Yifan and James have a very nice facility at Wollongong. And the University of Sydney is also acquiring a dedicated MicroED machine. And finally, Cam, I hope we can then lead the development of the method and then make the method available to the Australian community. So thank you very much. And also thank all the funding agencies and my collaborators. Thank you.
Thanks very much, Hong-Yi. We might wait and keep the questions until the end, just so that we don't get pushed too far off time. So without further delay, I'll pass over to Wim.
Okay, let me share my slides.
We can hear and see your slide.
You can see my slides? Okay, great. Let me get the list point. Okay, so good morning and good afternoon. And my name is Winjo, and supporting our APAC team for pharma business development. And Hong-Yi has a very good presentation. I think he almost covered most of the message that I want to deliver. So maybe I can accelerate a little bit. Okay, so 3 parts here. And after my section, I think I will have two MicroED experts to further cover the technical part and workflow. Okay, let's start from here. So before I jump into the MicroED, just let to start from the cryo-electron microscopy. So many of you may know the cryo-EM is an imaging technology that have been applied to solve, for example, protein structures, to characterize the nanomedicines, and also to understand the protein dynamics in the context of cells, for example. So today, the cryo-EM methods that we are going to focus actually is MicroED. I think Hong-Yi has given a very good introduction about how the MicroED work and about the diffraction patterns. And thanks to the advancement of the cryo-EM technology as a versatile tool, so in the industry, and actually we have been asked a lot about what's the situation in the industry, because cryo-EM actually starting from academia. And for industry, we more care about ROI, return on investments. And actually, due to the versatility in multiple applications for cryo-EM, it has been widely adopted by pharmaceutical industry. So here we can see the top 25 pharmas, 100% of them have been using the cryo-EM. And even for the next 25 pharmas, at least 50% are using cryo-EM. So these data are from the public available informations. So may have something we haven't got it. But you also, there's the other one that we want to show is that, so in the past few months and years as well, and we have been working very hard with opinion leaders, the industry leaders, and to introduce how the industry are using and start to adopt cryo-EM in-house as well. For example, Sanofi, Merck, and Genentech, and I think there's early as early as 2019. So we have a webinar with them and to introduce MicroED for pharmaceutical applications. And for small biotech as well, general biomedicines, and they explain how they use the really synergize the cryo-EM and AI to accelerate the drug designs. And Hongyi also have mentioned, so for MicroEDs, there are kind of different type of samples that can be suitable for MicroED. Chemical samples to solve the structures of the small molecules, and protein samples and nucleic acids crystals as well. Go to the next. Oops. So for why we care about the chemical structures, and in pharma industry, I think Hongyi also have a good explanation there. And the MicroED can be used as the pharmaceutical analytical tool to solve the structures and to understand the absolute configurations, the structure isomers, also the polyform situations. And also very important is the solid formulation to understand the crystal lattice, even to change the property of the pharmaceutical drugs as well. And here is just some examples for the US FDA to highlight the importance of the solid polymorphism situations and in the CMC stage. And have mentioned earlier, so Genentech is one of the pioneers in the industry to use their cryo-EM systems for MicroED work. I think the Hongyi also mentioned about AstraZeneca in this area. And why MicroED? There are many reasons, and one of it is for the less radiation damage and low electron dose, and for sensitive samples, and compared to X-ray, also compared to single particle cryo-EM. And the other reasons for biological micromolecule samples is the size of the samples. I think we are all very familiar with the micrometer range of a crystal that is very suitable for X-ray, but when it comes to microneedles, needles, and the precipitations, I think that will be very challenging for the X-ray diffractions, even for the micro focus beam in synchrotrons. But for the MicroED's technology, now we have seen more and more examples on this very challenging type of samples and can solve to near atomic level resolutions. And then I will just separate to two parts. One is for small chemical compounds and the other part for the biological micromolecules. So a simple workflows. So for small chemical compound determinations is a little bit easier compared to biological samples. You get the powders, crash it, and load it onto the grids and get the diffractions, and there's a series of software can be used to solve the phase and the structures. And as mentioned, the Thermo Fisher Scientific had developed the software EPUD for the automated data collection and crystal screening. I think our colleagues later will give more introduction on this software. And compared to protein crystals, and chemical samples seems to have much higher success rate. And for industry applications, and here is the statistical data analysis for NIS to highlight the robustness of this technology. And also, we have talked about the less electron damage and less restriction by the sample size. Another advantage of MicroED is we can apply the heterogeneous samples onto the grids. So you don't really need to purify your samples in terms of small molecule compounds, and you can just put it on a grid and identify the different structures also to understand the polymorph situations. And MicroED also played a critical part in natural product structure determinations. And one the traditional method like NMR has challenged for some difficult structures like what is shown here. And also, this is another example how MicroED complements the NMR in revising the complex structures of bacterial metabolites. And for example, this very complicated chiral centers and will be quite biased using NMR technology. But as a complementary approach, using MicroED to combine with NMR, we can have better understanding of the structures. And the last section is the biological micromolecules applications. And even though chemical compound structure determination by MicroED is more mature in a certain extent, but we are seeing more and more successful cases involving biological samples in public database. For example, the peptides, filaments, membrane proteins, ion channels, and also the viral proteins, protein-protein complex, antibodies, enzymes, DNA. Actually, there's already increasing numbers of success stories from here. Again, why there's increasing interest in using MicroED for protein crystal structures. And I think let's go to the nature of the situation for crystallography. I will consider crystallography as the black box. So you optimize your contrast, you try different conditions, you screen different additives, but at the end, you may still counter these problems. Very small crystals, needles, or precipitates, that would be very, very difficult for X-ray. But actually, under the electron microscope, actually these are not small anymore and is large enough for MicroED to handle. And here just a case from the ion channels. So you see a light microscope. Under the light microscope is precipitates. I think as a crystallographer, if you are, and you will see these problems a lot. And I mean, 99.9% of the time you will just forget it and try to optimize it. But under the microscope, and actually there's a case, and they can solve to 2 angstroms, and they can even see the waters along the ion channels. And there's more examples, and for example, tau peptides, and they try to different fragmentation methods, sonication and peptide and peptiding and vortexing, and different fragmentations, and trying to break down the microcrystals to smaller crystals, make it more suitable for MicroED on the cryo-EM systems. And as Hongye just mentioned, if your crystal is huge and there's the other methods like cryofib and milling, and this method has been under developing as well, and to make suitable size for the MicroED on the cryo-EM systems. Here is the example for the Golden Lab, and it's a GPCR examples embedded in the LFCB systems, and they use the film millions to make a really nice laminas and get the structures at the good resolutions. We have more examples. For example, the first novel structures solved by MicroEDs, amyloid fibrils, and very small crystals, and it was able to solve at 1.4 angstrom by MicroEDs to really understand the protofibrils and how it built to the filaments, and to understand the biological information behind. And you also have an example for the HIV inhibitor complex. So it's an inhibitor to inhibit the GAG cleavage. And it's critical to understand how the drug candidates bind to the targets in order to understand the mechanisms for further developments. So here is the case for the HIV maturation inhibitors, and also very small crystals, but it was able to solve by the MicroEDs at high resolutions, and you can visualize the compounds and to stabilize the GAG lattice. And we also have the other example for the enzymes, protoglobin. So it has been recognized as a good candidate for biocatalysis systems. So scientists would like to know the structures and to better engineer the enzymes to enhance the activities. And last but not least, so during the drug formulations, so there's a critical part to really to optimize the pharmaceutical properties. For example, the long-acting release, the superior stabilities, and also the low viscosities, for examples. But as limited for the X-ray diffractions, not all the crystals can be diffracted properly under the X-ray. So MicroEDs has been played a role here, and to really understand the different crystalline forms of the drugs for the optimizations of the pharmaceutical properties. So that's my part, and hope it gives you an overview of how the industry using the MicroEDs. Thank you.
Thanks very much, Wim. Boyin.
Hi, Ryan. Thanks. I'll share my screen.
Wonderful. We can see it now.
Thank you, everyone. My name is Boyin, currently based in Melbourne. So I'm an application scientist looking after cryo-EM applications as well as large volume analysis in ANZ area and also other areas of the APAC. So it's very nice to be here to share some of my experiences with experts in the community. So my talk will be very hands-on. I'll be focused on some details in sample preparation and data collection to help you improve MicroED data. So first we got some a few methods available for cryo-EM and just mainly for cell biology and but with subatomic averaging you could do structural biology, but the resolution still can't match single particle analysis, which is the next one. We also got electron diffraction method, like 2D electron diffraction, which was very popular when cryo-EM was still blobology, and then later was slowly taken over. And we got 3D electron diffraction as well, including MicroED. So MicroED sort of fill in the resolution gap in the cryo-EM techniques. So for single particle analysis and cryo-tomography, we can't really do very small proteins, just simply because the imaging is a low dose imaging, and then we don't really have much contrast for small proteins. But MicroED is actually quite good at solving smaller proteins as well as small molecules. It sort of fill in the gap and also but provides slightly better resolution than single particle analysis. And some benefits from diffraction. Diffraction pattern is invariant to stage drift as well as beam-induced drift. So but instead, cryo-EM actually you have to worry about sample motion and beam motion, beam-induced motion, so that you have to worry that the motion has to be fall in the threshold of motion correction. And for diffraction, for this method, you can also do low dose diffraction. You got high resolution and high throughput data. I'll just break down the workflow. Hong has already explained the workflow. So basically, for protein crystals, we do vitrification of the protein and then put it in the ETM and collect data. But sometimes when the crystals are too big, we use other mechanical methods to grind the crystals to smaller sizes, or sometimes we use a method called cryofilm milling to create a thin crystal lamella. The sample for the crystallization part, you could use the same setup as X-ray. So small crystals are more likely to form, but the difference is that for MicroED, you're actually looking for very tiny nanocrystals like these little ones, which is very different from the X-ray crystals we're looking after. And this is the example on the right is also has an interesting story. So the authors didn't really. Authors was able to grow very big crystals for the neck ion channels when they were in another institute, but after they moved to Genelia, even with the same setup, they couldn't grow big crystals. They keep getting this greasy slurry, but they didn't give up. They actually gave MicroED a try, and turned out that they have a very good cubic microcrystals there, which is perfect for MicroED, and they were able to solve the structure down to 2.5 angstrom resolution. So if especially when you're membrane protein people, if you have crystal like this, don't give up. Maybe give it a try with MicroED. So when the crystal is too big, there are a couple methods you can do to downscale the crystals. We as already talked about those. So you could do vigorous pipetting to do grinding, vortexing, sonication. The first four methods pretty straightforward. So I'm gonna spend some time with the required feed milling part to give you some details about how to do this. But before I talk about feed milling, we'll go to sample vitrification first, because you need to still freeze your sample before the cryo feed milling part. For vitrification, one of the options is VitriBoard. Same setup as SP and cryo-Thermo. You have a temperature control and humidity control chamber. Load your sample to the grid in the chamber, and then blot with the double blotting pads, and then plunge them into freeze. So you can see that from the movie, you load the sample to the grid in the chamber and blot by blotting pads, loaded with blotting paper, and then salvage your grid. So very similar to SPA. So we have some recommendations for VitriBoard. You have two options for VitriBoard, double blotting and back blotting. For double blotting, of course, as you all know, 99% of crystals will be blotted away, but sometimes they may leave some good microcrystals behind. You never know. For back blotting, we can. This is actually for samples with much lower concentration, so that you want to keep crystals on the grid. You can do this by disabling the front blotting pad with some hydrophobic film like Teflon or par film. In that case, you have to do in order to make a thinner ice, you have to use a very strong blot force and very long blotting time, at least 12 seconds blotting time. And even with back blotting, some crystals still got sticked on the Teflon. So still need to be careful. And it have other options like a GP2. GP2 is specialized in back blotting. It gives you a better back blotting results, but some good microcrystals may be blotted through the foil holes from the back. So basically, both Leica and the VitriBoard have pros and cons. So it really depends on concentration and size of your crystals. So we can't really give a clear recommendation about this. And so you got a cryo-feed technique to make smaller crystals when your crystals too big. This method is very similar to the on-grid lamella milling in cryotomography. So I stole this movie from cryotomography to show you how this works. So we currently have mainly have two products available for this work: Hydra Plasma PFib and Aqueous Tube Fib. They both have cryo stage and can do cryo work. So this is how it works. So you have what you are looking at now is the fib SCM chamber. You got SCM column, ion column, and sample can be transferred through the quick loader onto the cryo stage. And firstly, you can coat your sample with a protective GRIS layer to protect your sample from ion beam emitting. Protect from ion beam damage, and then find the coincidence point for both electron beam and ion beam, so that they look at the same structure, this same location at the same time. And then, for example, if this is a crystal, then you set up to move to a milling angle, usually about 10 to 20 degrees milling angle, and set up two counting windows with lamella in between, and then gradually do rough milling and milling, gradually using smaller and smaller beam to create a very good lamella. So the procedure is exactly the same as cryotomography. And then this slide, I want to talk about some differences between cryotomography. So basically, this is how a good sample looks like for sample. You need to have some buffer there to stabilize the sample, but in the meantime, the buffer can't be too thick, otherwise you spend lots of time milling. And then you can produce a very good lamella. And under the TEM, you can see the different layers. You can see a protective platinum GIS layer, the crystal itself, buffer, and carbon. You can diffract directly on the crystal, and then you get very good, very sharp signals because the excessive buffer has been removed. For the thickness, we actually recommend for 200 to 300 nanometers because it's quite different from cryotomography. Cryotomography people really actually aiming for thinner lamella because they think, with a thinner lamella, they get better contrast, and especially for people trying to solve the protein with subatomic averaging, they trying to get down to 100, 150, or even 100 nanometer lamella. But this is not the case for MicroED. For MicroED, you have to at least have, as a rule of thumb, at least 10 unit cells in the lamella so that your sample is isotropic. Otherwise, with a very thin lamella, you get very streaky diffraction pattern, which is not for data processing. So basically, we want to choose 200. In our hands, 200 to 300 nanometer lamella is the best. And we actually have the automation software to do this work. It's also called AutoTemp Cryo. Has been used by cryotomography community for a couple of years, and we have very good feedback. You can see that in the software you have 3 steps. Preparation step is for finding eccentric height and milling angle automatically, and then milling step is for rough milling, and medium milling thinning step is for polishing. So this software is on the right hand side. You can see there's a recipe there. As long as you have good recipe, and then it's a completely walk away acquisition. And also you only need minimal training about this because it's different from cryotomography, that you have to spend lots of time optimizing the recipe so that you get very thin lamella for tomography. For MicroED, you don't really care about the thickness too much. Anything between 200 to 500 nanometers, okay. So so you can just use a default factory recipe, and then you easily get lots of MicroED lamella quickly with very, very minimal training. And another thing that we want to use CryoFib is that it's a potential solution for preferred orientation problem. This example is from Hong Yi's paper published in 2019. He studied a protein called in a crystal called R2 logs. This protein crystal has a very flat plate-like shape. So after blotting, and all these crystals are kind of lie down on the carbon film, and then maintain the same orientation. You can see their data here. They actually achieved the 50.9% completeness from one data set, but after merging 21 data sets, the completeness only increased by 12%. So you can see that the preferred orientation is the main obstacle for them to getting a high resolution. So one of the solutions to overcome this our preferred orientation is to try to collect high TILS data. But unfortunately, microcrystals are usually too thick for TEM at high TILS. So now CryoFib come to the rescue, so you can actually prepare crystal lamella with different milling angles, and then try to get information at different tilt angles. For example, if you're aiming to collect 80-degree tilt range, and you could create a lamella with 20-degree milling angle, and then collect data from. It really depends on how to load, which way you load your data. You can collect from -20 to 60 or -6 to 20. In that case, you cover the 40-degree to 60-degree range of the reciprocal space, and then you can even use a higher milling angle to create a higher tilted lamella to try to get different information from different angle. iFilm is another hardware on the CryoFib. It's in-chamber fluorescent microscopy initially designed for cryotomography work. So, in cryotomography, the cells are embedded in buffer, and also under SEM, there's no contrast. You can't really find your target under the SEM. So what people do was to fluorescently label the proteins and then do some clamp in the SEM chamber and find the target to mill. And the community actually found a application for MicroED with iFilm as well recently. The paper just published last year, and there's also a few manuscripts available, which was just published this year. This is very useful in crystallizing LCP. So, for example, GPCR. But LCP is very viscous and thick, and extracting crystals from LCP is very difficult. And converting the LCP to the sponge phase, could make it more fluid, but it might damage the crystals. So in this case, iFilm helps locate the crystals and milling unconverted LCP. So what the authors did was to fluorescently label the protein before crystallization, and then mill use plasma fib to mill down the crystals, and they already have lots of success in samples like GPCR receptor, MP20, and human vasopressin 1b receptor. So this is an example of the GPCR receptor embedded in the thick LCP. And after you can see on the grid, there's completely no topology to tell you where the crystal is. But after you switch on the iFilm, you can see the glowing crystal here. And then you can aim for the area, and then iFilm also gives you the Z, the precision in Z directions. Then you'll be able to find where exactly the lamella is. And then you can see that after the meeting, you switch on the iFilm again. You can see there's a piece of GBCR crystal here, and then you aim for the crystal here on the lamella, and you get very good diffraction. That was sample prep for protein crystals, but if your samples is dry crystals or powder, it's much easier. Preparation is much easier. Hongye has already talked about this, and I just want to stress on sample loading options here. So for this kind of samples, you can actually do them at room temperature. It's just that the sample will going to be more dose sensitive because no cryo protection, but it works. Or you can just cool down in liquid nitrogen quickly. No need to use liquid ethane because there's not much difference there, but just need to be careful with ice contaminants in the liquid nitrogen, especially when your crystal looks very similar to ice crystals. You're going to have to spend lots of time in screening. And or you can also choose to cool down in the column. Just load the samples at room temperature and let the sample cool down in column, but it takes very long time to stabilize. But the sample will be cleaner. Now let's talk about some data collection. As Hongye already mentioned, that the MicroED is actually, in the MicroED data collection, the sample holder is actually doing continuous rotation. It's not like tomography that you do tilt, stop image, tilt, stop image. It's a continuous rotation. The holder backlash is minimized. And then you use a faster camera to do a continuous exposure, and then do a continuous exposure here, and you got a series of diffraction images. In that case, for every exposure, you're actually sampling a wedge of the sample. That's why the MicroED diffraction looks sharper and gives you a better signal-to-noise ratio because it's sampling a wedge of the sample. And it's a low-dose imaging. Overall dose has to be maintained roughly between 1.5 to 10 electrons. Sometimes can go up to 25 electrons. Really depends on your samples. And data collection options, if you don't have Thermo Fisher products, and then in the early times when Gonan Lab started MicroED, they developed an external device using a potentiometer to control the stage movement, or you could also use cryo-EM, such as the CR move script to control stage and camera exposure. When you have Thermo Fisher products, and it also depends on if you have MicroED package or not. If you don't have a MicroED package, you could use a Delphi scripting tool to control the stage movement, and use the camera rolling shutter mode and collect data in TR software. And also, but you need the FEI tomography software available for tomo alignment and optimized position. And without MicroED package, you can use 50 micron C2 aperture plus a 10 micron selective area aperture, because 50 C2 aperture actually gives you lots of scattering signals. With MicroED package, the situation will be more easier. So you can use EPUD automation software for data collection. You have a CTD camera for which movie mode is available. And you have a smaller beam stop, fish-only beam stop. And you also can use a 20 micron C2 aperture. In that case, 20 micron C2 is the limiting aperture. You don't really need a selective selected area aperture anymore. 20 micron C2 already gave you a very good signal. So overview of the MicroED package, you got CTD camera, and then EPUD software, smaller beam stop, fish-only beam stop, and smaller C2, and also a different lens series, so you don't have the rotation between different magnifications. So just quickly go through the MicroED package beams. So the beam stop on the left was a regular beam stop. You can see that it's very bulky. Also has very bigger stick here. And with the fish-only beam stop, it's much smaller, and also it didn't mask the low resolution signal. This is also part of MicroED package, the 20 C2. When you don't have the package with the 50 micron C2, you can see there's lots of scattering of the signals here. And you need to add another additional SA aperture to mask this scattering signal. And that includes more operation and more alignment. So with the C2 aperture, you just need a C2 aperture. You got very sharp signals, very good signal to noise ratio signals. And the C-type D camera is optimized for diffraction. It has a thicker scintillator, layer, which has very high sensitivity for low dose diffraction, and then also has a movie mode for continuous tilt diffraction. And then CDS is also available. You can compare see the comparison between CETA-D and CETA camera. You can see that performance of CETA-D camera at high resolution is much better than CETA. So for data collection, this is the workflow. So after you've got your cryo-TEM grid with nanocrystals, and first you get atlas, and then find the good grid square, and then get the image of the grid square, and find the crystal you want the image, and then increase the magnification, and then find exactly where which part of the crystal you want image, and then insert the 20 micron C2 and aim for the crystal and get diffraction pattern. And you really need to calibrate the image shift between all these magnifications so that you don't lose the crystal, and also minimizing the exposure. And after that, EPUD has a screening mode here on the right-hand side. It's actually a navigator. You go to the crystal, find the eccentric height, and then save the position, and the EPUD will save the X, Y, Z position for this crystal. Z position is very important. It's actually save the eccentric height for the crystal. And then after you press start, EPUD will go to the crystal one by one and take one image and one diffraction image, and for you to decide later which one to throw out and which one to keep. And then after you selected good ones, and just set up your tilt strategy, and then start collecting data. This is a video for the EPU workflow. Firstly, it's exactly same as EPU, so you can do atlas. Also, you can also do batch atlas in the screening tab. screen 12 grids all together. This is just exactly as EPU, and then find a good grid square, and find the crystal you want to image, and increase the magnification to search. And then find the crystals you want to image. I think this is just example. I don't think this is a good crystal. This looks more ice. But for the demonstration purpose, and then you can right click and move the stage to the crystal. And then go up, go one magnification up. Go to next preset, image acquisition preset. Usually we use 20,000 to 30,000 magnification, and then center the crystal, and then take a diffraction image for screening purpose. This is to center the crystal again, and then go to diffraction acquisition. And then EPU actually, once you switch to diffraction pattern, EPU will insert the beam stall, put a switch to diffraction pattern automatically, and leave the screen automatically, so you don't have to do anything. It's very easy to use, and set up exposure time, and press acquire preview, and then you get diffraction pattern for screening purpose. Okay. So this is a bad one. And batch screening in EPUD, so I've talked about that. So this is a new feature for EPUD. You can you got the navigator here. I've added more than 50 crystals at a time, so the software didn't crash. So so you can, it's pretty reliable. You can just keep adding lots of crystals here, and then start the automatic screening run. So, and then check the diffraction pattern later, and then decide which ones to keep. For the preparation before you do data collection, of course, some alignments need to be done. The diffraction alignment, Thermo alignments, and then which is the camera lens calibration is also very important to calibrate the pixel size. And then for MicroED, we can't really use the setup for SPA because we actually use lower dose for MicroED. So you have to lower the gun lens, and for example, for Technite column, use gun lens 8. We usually use gun lens 8. After you change the gun lens, you need to do gun alignments and save a FAG register, and then find the parallel illumination, and then also fix the diffraction lens stigmation. Measure the dose rates usually between 0.05 to 0.08 electrons per angstrom square per second. Overall dose may usually between 1.5 to 10, sometimes can go up to 25 depending on samples. Spot size between 8 to 10 on Technique column, and also need to do the optimized position calibration. Optimized position is to align the optical axis with the tilt axis so that they perpendicular to each other. If they are not perpendicular, you have some image shift here. Even if you're on your central height, your crystal will slowly drift away after when the stage tilts. So another thing is to decide your camera length for the sample. So, what is camera length? Camera length is actually the distance between the specimen and the detector. But on the TEM, we can't really, it's just physically impossible to change the detector up and down. Sometimes the camera length can be 3 meters. Just no such room can fit in such a TEM. So instead of changing the director position, we actually just the detector stays there, and we change the lens current to sort of creates this zoom in or zoom out effect to achieve the similar, achieve the effective camera distance. So this is equation for the camera distance. This is actually derived from the geometry between L and R, and then Bragg's law. Lambda is wavelength of incident electron beam. L is camera length. You really care about, and DHKL is the real lattice spacing. It's a known value for your crystal, and the R is the reciprocal spacing on the detector. So this is the equation is also used for calibrating camera lens. We usually we use cross grating, which is polycrystalline gold, to calibrate to use this equation to calibrate camera length. You can see from the equation that the bigger the camera length, the bigger the R, the bigger the reciprocal spacing, meaning that if you use a longer camera lens, then you're going to lose some of the high resolution diffraction patterns. So in order to illustrate this difference, I'll just put an example here. This is an example of aqua pouring 2D crystals. So with a camera lens 4,000 millimeter, you can see that you have very separated diffraction patterns, but your resolution cutoff is only 10 angstrom. You're not going to get very good resolution for this diffraction. And then if you lower the camera lens to 3,000 millimeters, you got more diffraction patterns here, and resolution cutoff is 3 angstrom. And if you keep lowering the camera lens to 1,750 millimeters, your sample can diffract. You can see it can diffract up to 1.5 angstroms, but but all the diffraction spots are squeezed or packed into a very small area. It's going to create some problems with indexing later. So this is the camera lens is all depending is all a balance between the indexing results later and the attainable resolution. For example, in this sample, 3 angstrom, a 3,000 millimeter is a good compromise. So you need to decide the camera lens for your own sample. And another thing you need to consider is the tilt speed and tilt range. So in general, the larger the unit cell, the finer the increment step needs to be to prevent diffraction spot overlap, because you're sampling a wedge of the sample. For a protein crystal with a bigger unit cell, there's so much information in there. So sometimes if you tilt too fast, you're gonna have overlapping information. So in that case, you have to slow down. So basically, we have some general. This is very general suggestions for small molecule camera lens 0.5 to 0.7 meters, tilt range plus minus 50. You can tilt faster, 1 degree per second. With protein crystals, a longer camera lens, because you have so much information there. You want to separate the information, so you have to lose some high resolution signals. You can use camera lens 2.5 to 3.5 meters, tilt range plus minus 30, 0.50 and tilt slower. This is for FEI or Thermo Fisher instruments, only apply for Thermo Fisher instruments. And tilt range is actually limited by overall dose. For example, for protein crystals, you can't really tilt to a very high range because the tilt range is limited by the overall dose. And lastly, don't forget that MicroED is very versatile. It can do more than proteins. It has applications in material science like metal organic framework, minerals, functional materials, etc. This is something we did with our training customer on a type of mineral. Can't say too much about sample, but applied MicroED on the mineral sample, and we got very good diffraction. And this sample is also not dose sensitive. In that case, you can use bigger tilt range, and you can do much slower tilt speed and use much higher dose rate. So you can probably get very useful stuff from just one data set. And then the only thing you need to worry is to not to saturate the detector, because you don't care about those, and you don't care about those now. So you have to make sure don't saturate the detector. Then the FEI TFS software can easily find out the detector was saturated by just through the TR software. Okay, that's all I want to share today. Thank you so much.
Thanks very much, Boyan.
That's all. Thanks.
Thank you. I think, again, we will just make sure that we collect the questions at the end, and then we'll try and answer them all. If not, we will respond to everyone and send through answers via email following the presentation. So next we have Yufeng, who will talk about processing of data.
Share my screen. Okay. Hello, everyone. Yes, lovely to join this workshop. I will mainly talk about data processing and a little bit structure determination. And Hongyi and Boyin has talked great about workflow, so I will only mention the different bit we are doing. Here's where we are. So we are in the Molecular Horizons Research Institute on the ground floor, and quite a new fancy building, only opened 3 years ago. So here's our workflow. The top part is for small molecular structure determination, and the bottom half for protein. So for our workflow, the data collection and the data processing are quite the same. The difference is at the sample prep and the final structure solving. So currently our workflow is quite mature with a good diffraction, with a good sample, with good diffraction, we can quickly solve a small molecular structure with good quality. For protein, the bottleneck would be still the sample prep. So every protein is different. This means for every protein, we have to test, repeat, and find the best conditions. So for data collection, we have a little bit different because we use all the regular equipment. We use Telos Arctic microscope. Here's where we store the data, part of our supercomputer system. After the crystal screening, the data collection, we also use continuous tilting skill for data collection. It's just we don't have the automated collection software, so we all do manual work. And all the hardware are normal, like the detector, we just have normal theta camera. So most great MicroED team use theta D camera for diffraction purpose. And for this tilting, we use a scripted software called Delphi. And imaging just use TIA. The TM imaging analysis software is very basic software. Give us .ser file. Really, yeah, not specific for diffraction work. But with our great teamwork and lots of effort, so we set up this workflow. Now we can collect really good quality of data with this all the settings. Small molecule, Boyan just recommended we do similar thing. We normally big range 120 degree. And for proteins, here's our protein image from last year, special lysozyme. So similar thing for macromolecules, we just adjust the tilting angle and integration time to get the best data before their crystal dies in the radiation. Talking about collecting tools, I think Hongyi also covered, and we have there's some choice of automated software, and what we are doing is manual work. And the good thing is normally we quickly process data and then adjust data collection strategies. But it's really, really time consuming, like 1 hour normally collect 10 good data sets. So it's really time consuming. So we would recommend for high throughput and training purposes, EPUD by Thermo Fisher seems to be really the best choice. Okay, so here's just a little bit introduction of our workflow. Now let's have a look at data processing and structure determination. So I plan to talk about 4 things, main things. and share some thoughts about the future. The main talk would be the data processing. So we use diodes for data processing. For data processing, for any crystallography, the diffraction geometry is the most critical thing. So with a good geometry modeling, we can get the very good model for our experiments. So for X-ray, classic X-ray and the electron diffraction are quite different. X-rays are more like waves, electrons are particles. But once electron pass the diffract part, they also generate wave-like property. So in theory, they all diffraction. Is for X-ray, people care about this angle, about the beam intersect with the detector. So this all based on the Bragg's law. Here's the classic Ewald construction I want to mention a little bit. So this based on the Bragg's law, so lambda equals 2d sin theta. So lambda is the wavelength, d is the distance between the atom planes related to the resolution. So here's the incident beam, diffracted beam. So this angle is 2 theta. So this a thing to consider. So in order for the diffraction reciprocal points to be captured on this sphere, so it has to satisfy this Bragg's law. So, and we call this sphere Ewald sphere, and with a radius of 1 over lambda. So in experiment, we would rotate the crystal, try to get as much as the reciprocal points on the sphere surface, so we can get a good structure. So what's the difference then between the X-ray and the electron diffraction? For example, X-ray, a standard machine with 12 keV, wavelength 1.03 angstrom. So this 2 theta angle if spectrum vector at 2 angstrom resolution, the 2 theta is about 29.9 degree. And and the Ewald sphere would be a really complete ball, complete sphere, and with the detector very close to the crystal. But for electron diffraction, it's also diffraction, and it also has to satisfy the Bragg's law. So, but we have much powerful voltage, 200 keV, and wavelength is about, for example, the Arctic 0.0250 angstrom. So with same scattering vector as 2 angstrom, this 2 theta angle is only about 0.72 degree. So this means this angle is so small. This means if we want to build a Ewald sphere, the radius would be really, really long. You can imagine a tiny bit angle and really long to get a huge ball. So in real experiment, it's really the Ewald is more like flat, nearly no angle. So this means the geometry is quite challenging. So, and with the long distance, this long distance Boeing just explained is achieved by the lenses. It's actually is a function of the TM lenses. So the real detector is actually fixed. So how to model the geometry? So this is a big challenge. So diodes was developed to solve this challenge. So that's a great thing about diodes. Is the full name is Diffraction Integration for Advanced Light Sources. So it was started from 2011 by UK Synchrotron Center and CCP4 to try to develop this software. Later, it became an internationally collaborative open source project. We got this first edition released in 2016. Now we have 3.19 version. And it's really, they have a great team. It's like update every 2 weeks. So the function is just getting better and better every day. and it runs on Linux and Mac command system. It does have a GUI version, but I think people normally don't prefer it. It has lower power. So the command version gives us big power to manage the data. So when we process data, our purpose is to, we get our diffraction image, try to solve a structure. So we try to extract the information out from these images. So not just the intensity, not just really the structure, also have the imperfection also inside. So all the effort is try to get a good model with lowest deviation as possible. So here's the flow chart of diodes. It looks quite similar as all the other software, like the classic X-ray diffraction software, XDS, start from importing the data and find strong reflection spots, indexing, get the geometry, refine the geometry, integrate all the reflections, and scaling the data, then export for phasing and structure solving. All similar. And if we have similar quality of data through every software, you can get a similar quality of result. So the unique thing about diodes is really about the, for example, the fine spots, indexing, refinement. It look through the whole data set in one go. So like XDS might process like window by window. So the great thing is when we get to integration, nearly the final step, we already have a good model. And this, because integration is really computationally expensive. So with our cryo-EM computer system, we can do like integration in 1 minute. But if the computer is not good enough, it might take 20 minutes to pass this step. So let's go through the steps. I'll see how it looks like. So first, import the data. Diodes has been developed to be able to read nearly all kinds of diffraction formats, from X-ray diffraction data to electron diffraction data. And it covers all kinds of common used detectors, all the companies. So if ever you have data sets that can't be read, it's very easy actually just write a reading header and you don't need to change anything, don't need to convert data, don't need to change diodes. So it's very handy. This is the importing program look like. It gives us a result with an imported file with all the metadata for the images from beam orientation, wavelengths, everything in the import file. So here's an example of our data after importing. So after we do the importing, second, we need to find the strong reflection spots. So the diodes find spots program, it does a 3D analysis of all the strong pixels. It pick up all the peak pixels, the strongest pixels, and give us a 3D shoebox. So this from the shoebox, we can see the highest peak pixel and the centroid of this spot. And the algorithm currently is really accurate and give us high quality of data later. And it's especially effective for data from CTD camera. I think the team really work a lot towards the development of our industries, try to match. So we will get a file of strong reflection file, including all the information about the reflection spots, coordinates, intensity value, etc. So after find the spots, the next step is to do indexing. So it looks complicated, but it's a quite robust program. So through this indexing, it discovers unit cell, orientation of the crystal, and give us a initial geometry model. So it maps the spots to reciprocal space. And by default, it would do a search for primitive lattice, start from Swiss group P1. So And after get the reciprocal lattice points, it does a 3D fast Fourier transform to give us real space lattice. So you can just repeat until you get really good results, feel satisfied with. Here's the output look like. So we will get a space P1 unit cell and the root mean square deviation and how much you have indexed all things. So if you are happy, you keep going. Not happy, you can adjust and try to get a better result. So after the indexing, if you know your sample space group, you can use the space group to process. If not, so we can try to do a symmetry analysis through the Brevis settings refinement program. So this program performs a full refinement in all Brevis settings that consist with the input unit cell. It analyzes all the symmetry and normally give a suggestion of a better, higher symmetry point group. So with all the output metric fit, deviation, the correlation coefficient, and operation basis. So yeah, and especially I think the good thing about diodes we like is this Brevis settings viewer. We can see the unit cell directly. So after this, we do more geometry refinement. This refined program is also quite robust. It does nonlinear least square methods to the refinement. In the earlier stage, people do refinement with variant model, but now, we tend to prefer do the with distance fixed. So we fix the total to 3 distance. And so to avoid any drifting during the process. Otherwise, it might drift and then the unit cell we got would be not inaccurate. It does the support centroid refinement. This quite easy, this step. Then after finish all this geometry refinement, we would like to build up the final model. It does 3D profile fitting to get this model similar like XDS on this bit. So the pixels are mapped to the Ewald sphere by 3D profile fitting process. And the pixels are analyzed by background outline rejection method. So finally we get a normal distribution. So it's quite reliable. So these are all the major steps. So after integration, we would get a really good 3D model for the experiment. And so are we ready to solve structure? So before that, so we have to get the data scaled and corrected for the experimental impacts like radiation damage. So the scale process, this program gives us a big report about all the statistic results from our data set. From here, like we would pay attention to the multiplicity, completeness, signal over noise ratio, all the R values to give us assessment about the deviation and the correlation coefficient. So this step we normally process over and over to get the best result. I will normally go through every image. So normally for a data set, the first few images not very good, maybe, because maybe suppose not enough or something else. And the last few images normally have high radiation damage. So we don't want those data that would damage our result. And we will go through the whole output and use CC half to decide resolution cutoff. Diodes normally cut off around 0.3. And we also can use a CCP4 to open the reflection coordinate file to see if have a visualize about the intensity spread to see if it makes sense or not. And for small molecule samples, if you have really good diffraction, we could solve structure just from one data set. it happened for like paracetamol, this kind of easier sample. And most cases, we will collect as much as possible and process the data set, choose a few best one to merge together. So we will get much better the statistical result. So yeah. And here is, till now, I think I have gone through the major data processing steps. So I would like to summarize about some challenges. So the main challenge is the uncertainty of diffraction geometry. So there are many factors impact on the diffraction geometry. For example, the beam center. This one with beam center not correct. You see the reciprocal lattice distorted, and the right side one is correct one, beam center, very accurate. We get nicely line up dots. And for detected distance, same thing. Boeing has explained into quite detail. So we must get the distance calibrated before collecting data. And even after that, maybe when you process, you still want to have a look. If you know the unit cell from published paper, you can refine it even better. So rotation axis is very important. So if the orientation is not right, you wouldn't be able to index well. So also there's handiness issue. I think we have some audience from pharmacy industry. So the handiness for pharmacist samples would be really crucial thing. So for example, these black dots are nicely lined up reciprocal dots with a correct rotation axis. So for X-ray crystallography, if you use inverted rotation axis, so you would get some arced data and you couldn't process, so you wouldn't make such mistake. But for electron diffraction, as we mentioned, because the Ewald sphere is like flat, and even you use an inverted rotation axis to process, the dots still line up nicely. It just rotate to a little amount, rotate away from little amount, still line up nicely. We can still process the data and we can still get a structure in the end. But guess what? So the structure we get will be really inverted, so the wrong hand. So this is the thing to really need to be careful. Other challenges like we need enough good diffraction. Sometimes you spend a long time do the experiment, got some diffraction, you really want to get some result, but you could be able to process, but might not pass the integration step because we need enough number of reflections to be able to get a model. And other things, distortion also happen a lot, like lenses not calculated well, or rotation speed or something else. This image from my early stage experiment couldn't process. Okay, here's all the thing about data processing, the main things. So I planned to mention a bit about phasing and structure determination and give a bit example of our experience and share some insights. Also handiness issue. So phasing and structure determination for small molecules, we use shell X for direct phasing and OLX to do the refinement, get the structure. For macromolecules, we use Phoenix for both phasing and refinement. So the phase problem is crystallography problem. So it can't be measured directly like intensity, but we have all the tools and all the great software are widely known, so I won't go through details. So macromolecule structure, we use molecular replacement based on the evolution theory. Here's what the software look like for OLX2 for small molecule. It's quite easy to use and lots of functions give us really beautiful result. this one is one of the results we got. And I want to mention a bit about a lysozyme we solved last year. So we solved this lysozyme. So the crystal first, when I grew the crystal, I adjust the conditions, try to get nano-sized crystal for electron diffraction. So the crystal grow very short time and get nano-sized crystal. And it's solved eventually in Swiss group P21 monoclinic with this very unique unit cell dimension. So I first didn't really find a similar one in the PDV bank. There are at least more than 100 entries about lysozyme there. Then I talked to Hongyi, and Hongyi suggests, looks like a new polymorph. You can publish paper. Quite exciting. And later he recollected some group in Italy published a similar thing. We found the paper, and it's really similar. You can see all the parameters. So I'm really happy to know this. So this means our whole workflow from hardware settings to data processing, the whole process is really reliable. So we normally think because the challenge of the geometry for electron diffraction is hard to get good accuracy for cell parameters. But after all our experience, we think the truth is if everything is done properly, we can get really accurate result. So this really very encouraging. We feel very proud of our workflow, and we are ready to take more challenges. Okay, next, I think I would like to share more of the structures we solved and share some insight. This one we solved early stage, 2 years ago, carbamazepine with 0.85 angstrom, and acetaminophen 0.8 angstrom with really the electron potential map, really beautiful. Of course, we also had quite some samples from our real lab, from our colleagues. This sample was from Paul Keller's lab and by his student Safres Ailai. And we solved this structure to 0.7 angstrom. So this resolution means that all the hydrogens in white color, all the hydrogens can be determined with confidence. With this resolution. So till now, I have introduced the diode software and all the other software to eventually bring us to the structure. So for diodes, there are about 100 commands. So the thing I want to share is with this power, really we are able to deal with any tricky data set. As long as you have good diffractions, doesn't matter distortion or anything, we can always find some way to solve the structure. But I want to share the experience about my earlier stage experience. So we don't want to really mess up with the data. So here's the sample from Professor Paul Keller's lab. It's a sample in the middle of the research. They want to know what the structure is. So they give us 3 expected structures. So because the diffraction was not very good, I tried lots of effort, finally solved a result. The good thing is the part we want to know is quite clear about this side chain, quite clear. I got the side chain 4 atoms. Then I'm thinking, okay, maybe I try to refine it to get it, see if it can be a bit different, more similar as the expected one. But later, they sent it to a lab with a new powerful X-ray machine and determined the structure is actually the same as the original result. So this was really my first structure from real lab. I think the lessons I learned is let the diffraction speak. Don't try to mess up with the data. So more samples from Paul's lab, a second sample. So I want to give you an example about how different sample brains. So this first sample has good diffraction, but not really good enough. The second one is really good. So for the first one, I have tried a few ones, really, because diffraction not very good, we couldn't solve structure. For this one, it looks quite okay. I was determined to really get something out from it. I spent more than 3 weeks, did 3 rounds of experiments. The first 2 rounds couldn't solve anything. So the third round got a big success, but the data collection, I spent 28 hours, and data processing spent 30 hours. So the encouraging thing is the part the researchers need to know was quite clear. So, but with the second sample, because the diffraction was really good, so the data collection sample prep only took 1 day, and then data processing 4 hours done. We got the structure with very good quality, can be used for publication. So from all this experience, so we know the first important thing I want to let you know is really give us good samples with good crystalline material. And because I think all our speakers have been mentioned the difference between the how to get this out. Okay. So the difference between the X-ray and the electron diffraction. So the electrons with the every inelastic event, X-ray have high energy deposit, but electron have very low energy. The difference is like 40 eV compared to the X-ray 12 keV of the whole photon. So you can imagine why the electrons have diffraction has very low damage, much lower radiation, and much more efficient useful scattering but ratio to the radio damage. So that's why if you want a high resolution structure with a good sample, MicroED would be really good choice. Other advice would be if you have good big crystal, really you could try X-ray diffraction with much cheaper, and cryo-EM always have big cost. And try other ways first, like mass spec, AMR, everything. But for currently we have lots of researchers doing protein study in every different area. So if you have really challenged to grow large crystal, for X-ray crystal, normally it takes 1 or 2 weeks to grow large crystal, and this would be the time you consider use MicroED. And also, if you have small molecule weight of protein or peptide to study, MicroED also give you this choice. So currently, our single particle analysis approach, we can achieve molecular weight about 47 kilodalton. So bigger than this can go to our single particle workflow. If smaller than 47, MicroED is great. So just remember the crucial thing is a good diffraction. Okay, this is all the thing I want to share. And about the handiness, I think Hongyi had give some great introduction. I will just mention a bit about the information I want to share. So for X-ray crystallography, you might spend a whole life working on X-ray, but won't see the dynamic diffraction. But for electron diffraction, dynamic is really the normal thing because of the strong beam, the strong interaction with atoms. So how to get the handiness? With X-ray, we have the Fleck Friedel pair, Fleck parameter, but all this Friedel loss is broken with electron diffraction. So I found a few reports. This one was reported by nanoimaging team in America. They have a great team, did quite some MicroED research in the past few years. So this one, they use an internal chiral standard synthesized with the active pharmaceutical ingredient. So we can imagine in the end, because there's a known indicator, we know which structure is the correct one. And currently, the software package is the best choice. So this software by Lucas Platinos from Czech, and they have the Yana 2020 do the handiness refinement, dynamic refinement. This pass 2 for data processing. Because these two are developed together, they work together very well. So I talked to Dio's developer. There might be some concern, but currently Dios can export to processing Yana, just not as good as data processed from PES, because these two were developed together, of course, so they will work the best. Another way I noticed from material science colleagues, they do zone axis comparison, and also another way is to do the Friedel pair analysis in a single precision electron diffraction pattern. I feel this makes great sense to me, and we could adopt this for a process. So here's the main talk I want to share. And finally, I'm just try to inspire everyone with some thought about the future of MicroED. This is a paper published by Morogami just a couple of weeks ago. So he talked about MicroED has passed the first stage of development with both theoretical and experimental solid basis. Now he think we are entering a new era of growth and expansion to all the structure study labs. We totally agree with this, and we're really excited to see these areas growing so fast. Oh, by the way, this Mauro is the team leader for the Italy group who published same polymorph as the lysozyme we found last year. So it's really interesting. And so we are now ready to take more projects. We have been doing the study for antimicrobial resistance targeting the polymerase 3 beta subunit. So we have crystals grown by Erin Oakley's previous PhD students, and there's big challenge of this sample prep. We also have colleagues from neuroscience who are interested in collaboration, like then we have the epilepsy study, and Luke McEllery studied sclerosis for many years, motor neuron disease. Both projects have smaller peptides to study and related to the disease. So quite exciting. We might start collaboration soon. So for the future, so we would expect, I think in the near future, there might be a big jump of the software. So I think our industry and researchers have really close collaboration to bring the software to another really higher level, more automated and less challenging to training students. And this brings more challenge and opportunities as well to our industries. Thermo Fisher and other companies have been really creative and doing some initiative development on both software and hardware. So I think this whole field is really exciting. And finally, I would like to say big thanks to everyone who gave us great support during our journey in the past 3 years to develop the MicroED in Wollongong. Here's our team and our director, James Bauer, and he's a great physicist. And I want to say big thanks to Erin Oakley, our previous team member. Without him, this project might have to happen in the first place. He's a master in crystallography for 20 years. Also, big thanks to all the big leaders in this field in other countries. Tammy Goni was originally James worked with him back to America. So gave us great support at the beginning. David Waldman from Dios and Brent from Arizona and Hongyi also. We have great collaboration and really great, very excited. Hongyi moved here recently and we are so excited to collaborate together and do more great works. And I'm sure with Hongyi's help, our whole work at MicroED will go to an up level. Finally, a special thanks to our organic chemist groups. They give us great encouragement from the beginning and always give us samples to try to challenge us. So big thanks to Chris Highland, Stephen Penn, Paul Keller, and all the students involved. Thank you so much.
Thank you very much, Yufeng.
Okay, thank you.
James, over to you. Sam, we'll come back. If you can just before we start, James, Sam, if you can just type your question into the chat box, we'll address it when we're finished the presentation by James, which hopefully shouldn't be too long.
Thank you, Ryan, and thanks, Yifeng, Hongyi. Yifeng just makes it look so easy, doesn't she? So
yeah, hard work, 3 years.
So yeah, so Yifeng started with Aaron Oakley, Josh, about 3 years ago, and he did a really great job of getting her up and running, and she's worked so hard to really make this happen. So really amazing. And so what we have to present to you is this pipeline and access to it and the ability to do a lot of this stuff and get the training and everything you need from us. So let me just go ahead and share my screen here. All right. How's that look? Can everybody see my screen? Yep, fantastic. All right, so I'm just going to give you a little rundown about what we do at the cryo-EM facility at University of Wollongong, which is an openly facing customer support environment. So a customer-centric approach to training, data collection, big data processing in cryo-electron microscopy, and electron diffraction. So this was a $85 million investment by the University of Wollongong in about 2016. I came over 2017 to help design and build the facility. And this was kind of the brainchild of our vice chancellor at the time, Paul Wellings, and our previous director, Antoine Van Ohen, and Nick Dixon, who's the director of the Center for Medical and Molecular Biosciences at the time. So obviously what you've seen so far should really let you know that, wow, there's a lot of different angles. We've got physicists, mathematicians, engineers building microscopes, all of this, multidisciplinary environment, and getting people to work together was critical in helping us design this building. So here you can see kind of the original design of the building, which started in about scheduled to open in 2019. You can see the building site May 2018, which is when we took the parking lot and started to build this facility. And as you can see, July 2019, we're actually on target. So how often do you see a building like this go up on time? So what we did was spent a lot of time and money designing this building. So this building is built without any steel rebar. It's all fiberglass reinforced polymers. So we really went to every effort to meet those stringent Thermo Fisher requirements that just about nobody can meet. So we went for it. These are the rooms we designed in order to get really, really high performance. This is a big Faraday cage you can see welded together, which is what really needs to be done to eliminate the electric magnetic fields. We moved power substations across the campus, and here we are, building completed and ready to move in January 2020, so right on time. And this is us installing the Arctica. There's the machine sitting right there. Here's the team from Thermo Fisher and my team installing that into the building, and the cryos installed in the room on the buildings. This is the control room, so you've got to keep everything out of the rooms. You've got to build this environment which is completely clean of any kind of acoustic, electromagnetic fields, and put the people outside the room. So these are the instruments that we have on site. So we've got an Arctica, Thales Arctica that you see on the left. That's got a K2 and a Falcon 3 detector on it, and that's primarily the instrument we're using for microelectron diffraction. And we can solve structures on that in single particle down to 1.8 angstrom now. The Krios is in the middle. That's primarily used for all really high resolution single particle reconstructions. The T12 instrument, which you see toward the right-hand side, is what we use to train our customers. So this is actually where Yufeng started learning diffraction on this instrument. And this is kind of part of our workflow. We have these VitraBots. Everything's available to be booked and trained on. This is our workflow here, starting with sample prep, working our way through intermediate resolution, high resolution. We've got a whole section of local high performance compute that we provide to our customers. So that would be all of you wanting to do MicroED, hopefully, or single particle resolution. We also have remote access and secure data transfer protocols with our pharmaceutical companies in order to keep that data secure. And we've got tomography, single particle, MicroED solutions, all to get high resolution. Just Google UOW cryo-EM if you want to get access. This is something we use just to make sure that our machines are working properly. So apoferritin with the magic apoferritin sample from Thermo Fisher down at around 1.3 angstrom resolution. So atomic resolution, you can actually see how well we can do with this technique. So we do provide training on all aspects of this. So generally it starts with a 3-day one-on-one course, learning electron optics, then move into EPU tomography, automated data acquisition on our cryo systems, MicroED acquisition, operation of the cryo instruments. And then of course we provide all the infrastructure and the training to access all of the compute you need to run Reliant, CryoSpark, MicroED, which would be Dials, OX2, Phoenix, etc. And small group data processing on our local high performance compute. So really you don't need any infrastructure. We provide it all for you. We provide remote access to everything. This is one of our first initial collaborations with Thermo Fisher. This was our sample prep school. And here's our sort of first class there. This guy here, Pep, is now working at a pharmaceutical company. So people are out there getting jobs now. So this was kind of one of the first collaborations, sample prep, and a big reward for the best sample. In terms of our compute infrastructure that we provide to all of our customers, we provide the microscopes, the storage facility. So we've got 1.6 petabytes of local storage now on site. High-speed networking. We've got data storage duplication. We've got real-time GPUs running on the back end of these machines that tell us right away how well we're doing. And by the time you're finished acquiring data, we'll have a structure for you for our single particle work, local visualizations, and like I said, remote access, and lots of collaborations with RNet, Xenon, Globus, NVIDIA, and our storage partner, Vidura, previously Panasys. This is what our compute looks like. And we've got 2 real-time processing GPUs, a V100 GPU mini cluster, looking at mostly processing Reliant jobs. And a lot of this is going to be, that you've seen already, that the MicroED is going to be built into this workflow. Right now, we've got small machines, small workstations running the MicroED systems and managed by Yufeng, who can do a lot of the training on those systems. This is what the on-the-fly output looks like. So as you're acquiring data, by the end, when you're done, you have a structure, and almost immediately you can tell if your data is good enough to carry on. What we built recently is a 16 GPU mini cluster. This is devoted to our customers for remote access. So we store everything for you. We let you work on it remotely on our high performance compute infrastructure. And when you're done after 6 months or so of processing, or something maybe less, we provide a method using Globus to move all of your data back to your home laboratories. Of course, we've got single particle workflows, helical reconstructions as part of the single particle processing pipeline. We're now working on tomography algorithms. We have our first few tomography data sets. This is some old data from UCSD. Can't show you the new data, but we've got some very exciting stuff coming out now. Also, another thing that we're working on, so this could also be related back to MicroED stuff, is soft materials. In this case, this is looking at lithium polymer batteries and coming up with non-flammable solvents for the battery production. So your batteries don't catch fire. So this is taking cryo-EM techniques and applying them to material science, which I'm sure is going to be happening with this MicroED technique. We now have training, data collection, and processing on MicroED, both for small molecules and protein structures. This is our current system, the Talos Arctica, which you heard about some of the upgrades. So everything we've been doing is by hand, really with chewing gum and duct tape and pushing buttons on multiple softwares at the same time to get these data sets. Obviously, there's much better ways to do that. So we're looking at replacing the standard beam stop with the Fission beam stop, the small beam stop, and moving into EPUD software, which is going to make life a lot easier. Moving to the smaller 20 micron condenser aperture, potentially upgrading the lens series for MicroED, and maybe looking into the C to D camera. We do have a K2 on that system. We're still a little nervous about putting diffraction beams onto our K2 camera, even though we're told it's okay. And then access can be booked through our online scheduler. So if you're curious, go Google cryo-EM at UOW. This is book your own sessions, book your training, book your everything you need at the facility. Of course, we are also part of, we are a node of the ARC Training Center for cryo and electron microscopy of membrane proteins for drug discovery with Patrick Sexton at Monash University, along with all of these pharmaceutical companies you see here. So on both sides, single particle, tomography, and MicroED, there's plenty of applications for drug discovery, and we're trying to be right in the middle of that. So that's my plug for cryo-EM. I want to thank all of the people on our team. This is our cryo-EM facility, myself, Josh Silver, our IT and data solutions expert, Jason Andretti, IT and data management, Dr. Simon Brown, customer solutions expert, and Dr. Yufeng Xiao, who you've already heard today who runs the microelectron diffraction workshops and workflows. And I'd like to special thanks to Dr. Tamir Gonen, who spent a full day with me up in Sydney showing me how to do this technique, which we then adapted kind of through our duct tape and chewing gum methodology to produce what you see. So thank you for your time, and we are available if anybody feels like they want to try something new. These are various collaborations that we have across the country. See your name there at the bottom. Our collaborations across Australia, and of course, our plan to conquer the world. So that's it. Thank you very much. Really, really nice to have you all here.
Thanks very much, James. So I'd like to mirror and repeat James's comment about it being really nice to have everyone join us today. I think we have almost everyone who started the conversation with us a couple of hours ago. We might address a couple of the questions now. Does anyone have any questions that they'd like to ask in person on the call, or should we just look at the questions in the chat? We might start with the questions in the chat. From Will Lewis, Yufeng, can dials address incommensurates? You're on mute, Yufeng.
Okay, thanks. So I think this is really cool question. There should be some way to deal with it. Yeah, I'm not very familiar, but if you can, William Lewis, please explain a bit more.
Maybe I can add a little bit. So for in
Yeah.
commercial rates, modulated structure, so YANA is really designed for that. So PET's YANA is probably the better route to go to. Yeah, so YANA is probably the best software for doing this modulated structure, even for X-ray.
Thanks, Hongyi. And from David T, do we need to know the real camera length in diffraction to get the maximum resolution diffraction spots? Or how do we know the resolution that we're getting from the crystals? I think that question came before you presented on the dials software.
Okay. Yeah,
package.
I guess the presentation should answer the questions. So the camera lens, we need to get really accurate to be able to get the unit cell parameters correct. So for the resolution limit, so through the scale, dial scale, we see the CC half, the correlation coefficient among two randomly data sets, two half data sets. So we normally use a cutoff about 0.3. Yeah.
And we're also using gold to do a lot of calibrations right up front and get those camera lengths spot on. So
Yes.
Yeah. Thanks. I think we had a question from Sam Costi just before we started James's talk. Sam, do you want to come off mute and ask the question? No? Maybe there's some technical issues. Do we have any more questions here? No worries. Okay.
Yeah, I had a question, Ryan.
Oh, sorry, go ahead, Eric.
It's actually in it's probably been answered, but my question was really for powder and chemical drugs, can we use XRD or microfocus beamline to get the structure and the phase changes? That's maybe the first one for you, Hongyi. I know it's like kind of on the border, right? And there might be some stipulations around the sample, right?
Yeah, usually I think powder diffraction is probably still the best way to do bulk phase transformation. So my colleague actually built a in situ heating liquid cell for synchrotron, and they could follow the phase transformation during a porous material synthesis. But I guess for drugs, I think it's the same applies. If the bulk is changing to another bulk, then you can probably see it. But then if you have minor phases, it become difficult to detect.
Yeah, and what does that minor phases translate to in terms of the data, and how would it affect the development of the drug, the formulation, the quality? Like, what's the relation between the phase and the?
Yes, yeah. So for the drug part, I probably can't say too much, but usually people do this for IP protection, for example. If there are minor phases that has been IP protected, but otherwise, in terms of the crystallography methods, I think yes. So it's really just the composition of the powder that maybe research is interested in. How pure the phase is, right? So usually people trying to grow a compound or new material, they want to be phase pure. But how pure is this phase? Then we can give a number, put a number on this to see if there are any minor phases or undetected phases in X-ray. But X-ray is still, powder is still the best and easiest way to do phase analysis at the moment.
Thank you very much, Hongyi.
Q&A. not in chat.
Oh, sorry. Apologies, everyone. I didn't see this one. From Maeda Mahmoud, how feasible is detecting membrane protein structure with MicroED?
Yeah, there are publications about Thermo, I think, right? There are publications about Thermo last year about the membrane protein they solved. It's really like a breakthrough, because in the older days, we don't think membrane protein can be treated in this way. Yeah, please add more, Hongyi.
Exactly. So it's back to the crystallization problem. It's still crystallography method. So growing crystals probably is still the key. So if it's soluble, it can grow crystal, but if it's membrane protein, so most cases you don't have soluble membrane protein, so you maybe need to use the lipid cubic phase. Actually, we did that before, so it's easy to actually to turn the LCP into a sponge phase by adding some PEG. But then in our case, the crystal was slightly too large, so I think it was the same protein crystal Tamira worked on later. But again, to stress this again, it's really a crystallization problem. So if we can find a way to make very nice membrane protein microcrystals, then MicroED will be very feasible to do.
Thanks. So from Philippe Kremer, there are publications showing that there are benefits in using post-sample energy filter for clearing out the inelastic scattering, since the filter increases the signal-to-noise ratio at low K region of the reciprocal space. It becomes easier for your integration software to pick these spots and also decreases the need to use non-physical fitting parameters like XD during structure refinement. Sorry if I missed any examples, but have you tried using Selectris on MicroED experiments? Could you please comment on your experience with it?
Yeah, maybe I think some official people should answer. I think it has been tried already.
I think most of us are a little scared to put that. Most of us have direct detectors on the back end of our filter, so we're a little nervous about blasting that thing with the beam. But I don't see any reason why it can't be done. The K2 detectors have been used for MicroED, so it probably would work if you're brave enough.
I think we should have a discussion too, also from the Thermo Fisher side. Obviously, we'll entertain all different types of strategies, but we also need to help users measure that risk.
Exactly. Actually, I heard from Tamir. They've tried the Selectris with Falcon 4i with a great result. It's a great energy filter. They also said. But before us, we've tried the GIF with a CCD detector, even ultra scale. So it was also usable, but then aligning everything is a little bit time consuming, I would say. And how much benefit we get from the energy filter really depends on the questions we want to answer, right? If we want to look at fine details of the structure. And also, as you said, the EXTI structure during the structure refinement. So we don't recommend to use EXTI if we don't have to, because extinction is a very special phenomenon of X-ray, which doesn't occur in electron diffraction. So using this during electron diffraction refinement is just numerically to improve the refinement.
So again, from Felipe, how would you compare Lay's cluster grouping code compared to older software like Blend that predate MicroED and hence maybe use statistical figures that might not be useful when performing electron diffraction?
Yes, yeah, the idea is very similar. So two parts of the question. The first part, blend is just also for unicell parameter based clustering. The trick in blend is that instead of using 6 parameters like A, B, C, alpha, beta, gamma, blend uses the body diagonal. So it converts all these 6 numbers into one number to be compared for the clustering. I think Lay software does a little bit more. I think we look at all the 6 parameters. And also in Lay software, we have 2 levels of clustering. The first one is to do the blend, right, the unicell parameter clustering. And then the second round, we can also do a correlation coefficient of common reflections or the correlation coefficient between 2 data sets to see how much correlates the intensity itself comparing between data sets. And so with these 2 levels, we can have a better separation of the phases. Of course, as you said, statistically, MicroED, the unicell is not as accurate as X-ray, but in most cases, if the unicell deviates quite a lot, and it's quite simple and easy to distinguish them. And also this depends on how accurate our unicell parameters are from the data collection. But then the second part, the CC will have even more ambiguity in the clustering. But then this is for really look at something that has similar unit cell but very different structure.
So from Santos Panjakar, what kind of detector is used for MicroED? And what's the DQE of the detector? Is it CCD, CMOS, or pixel detector? I think the answer to this question is that it really depends on the specific application and the configuration of the instrument that you're using. We have cameras that are CMOS or direct detectors, but I think it's feasible that some of the hybrid pixel detectors will be applied to this type of technique in the near-term future. I think we answered David's first question about the camera length. What type of grids do we suggest for biological samples for protein macromolecules?
Normally, holy grids, any kind can try. Lazy or the standard, holy grids, I would say.
Yeah, I would say try the cheap ones first, but it really depends on the sample, as Yifan said. Those quantifiers are quite expensive, right? But we found what we found was if your sample grew in very viscous medium, like if your mother liquor is very viscous, then having larger holes probably is beneficial. So 3.51 grids is pretty good for these crystals growing viscous mother liquor. And then otherwise you can use the normal 1.2, 1.3 or lacy carbon grids also work, which is much cheaper. And recently we also found C-flat grids. So these very fragile grids, they actually conduct, they discharge a little bit better. So it's better for crystals if they build up charge. On normal grids, C-flat is something you could try also.
Okay, I think that's all of the questions we have. So once again, I'd like to thank all of our speakers today, Wim, Hoyin, Yufeng, Hongyi, and James. And just want to point out again that James's facility is open for external clients. So you can reach out to him at Molecular Horizons. We'll pass through contact details to all attendees afterwards. And thanks again for joining us today.
Thank you.
Thank you.
Thank you.
Thank you, everybody.
Thank you.
Thanks,
Yeah,
presenters.
pleasure.
Thanks,
Everyone.
Wollongong team. Thank you.
Bye.
Yeah.